And good afternoon. Welcome to Chapter 3 of Anatomy and Physiology. In this chapter, we're going to be talking about the cellular level of organization. So the cell is the basic unit of life. The cell was first identified by Robert Hooke with his microscope. He's the one who coined the term cell, and it wasn't until around 100 years later that Antoine von Leeuwenhoek was able to see living cells that were moving. He saw likely amoeba and other protozoans. So in this chapter, we're going to look at the cell structure and the cell's function. And we want to know or understand cell level because it's at that cell level that everything occurs. So cell is where metabolism occurs. The cell is the area where medicines will work. And it's at that cellular level that most diseases occur. So understanding cell structure and function is key to understanding human anatomy and physiology. This is a figure from your textbook that's showing a lung cell undergoing mitosis. So you can see the chromosomes right here kind of lining up at that metaphyseal plate. Or actually, they're starting to pull apart, I guess. I see that now. So they're lined up here at this metaphyseal plate, but do you see how these spindle fibers are moving away? That means that they're actually in anaphase. Okay? So the study of cells is termed cytology, and we weren't able to study cells until we had microscopes. And so the discovery of a microscope actually didn't occur until the 1600s, late 1600s or mid 1600s, with Robert Hooke and then Leeuwenhoek. A little while after Hooke. They both made microscopes for different reasons. Most of the cells of the human body are going to be larger than a micrometer in size. So, but they're smaller than a millimeter. So we measure cells in terms called micrometers, and is that abbreviation for micrometer, and a micrometer is 1/1000th of a millimeter. So this is a tissue that is composed of multiple cells, and tissues are made up of multiple cells, and this tissue actually comes from the lungs. So when we look at cells, we have three main areas that we want to focus on. We look at the plasma membrane, which I put the plasma membrane over here, so you can kind of see a plasma membrane, partial plasma membrane, I'll say that, composed of a lot of different parts. The plasma membrane is the barrier that also acts as a selectively permeable membrane, allowing certain things in and other things out of the cell. Then we have the nucleus. The nucleus is inside of the cell, so out here is that plasma membrane, and inside we have this nucleus. In general, all human cells have a nucleus, at least one. Most cells just have one. There are examples where we have some cells that have more than one. And we also have an example of a cell that has no nucleus, and that's the red blood cell, which we'll talk about in a little while. The nucleus is the region that contains our DNA, our genetic material, which is the blueprints that make us who we are. And then we have the cytoplasm. So between the plasma membrane and the nucleus, we have the cytoplasm. The cytoplasm contains the cellular structures called organelles, and the fluid called cytosol. So these organelles are kind of like the little organs of the cell that do the function of the cell. And they all float around in this liquid material. So in general, cells have to be able to maintain their shape, their integrity. They have to be able to take in nutrients, and they have to be able to get rid of waste. Those are basic functions that, in general, all cells are going to do in some form. Whereas there are certain types of cells that also undergo cell division. So cell division is the production of new cells, and this typically occurs through mitosis. So when we're talking about cell division, we're looking at mitotic division, where we're producing new identical cells. There's another type of cell division called meiosis that we'll talk about when we get into the reproductive system. And meiosis is a cell division where we're producing unique sex cells. In this case, we're looking at one cell dividing to produce two identical daughter cells, usually for growth and development, as well as for repair of tissues. When you have damage to your tissue, your skin is constantly sloughing off. You need to always produce new cells. If you have any damage, if you break a bone, you're going to have to produce new bone cells. So those types of cells are able to undergo mitosis, whereas other cells like heart, cardiac muscle cells, most of your nerve cells cannot undergo mitosis. You're born with the cells that you have for life. So let's focus first on that plasma membrane. The plasma membrane has multiple structures that make it up, but the most common structure is called a phospholipid. You learned about phospholipids in Chapter 2. Phospholipids are fats composed of a glycerol head and fatty acid tails. The phospholipid makes up the membrane, the plasma membrane, the majority of the plasma membrane. The glycerol heads are the polar regions that interact with the extracellular fluid and intracellular fluid. And then you have hydrophobic tails that interact with each other. So a plasma membrane is composed of two layers of phospholipids. Tails are sandwiched in between the heads. This makes it a structure that is known as an amphipathic structure. Amphipathic meaning it has charged and uncharged regions. So this is what the phospholipid head looks like, and here is your fatty acid tail. The fatty acid tails then are going to interact with each other. The glycerol head here is going to be able to interact with other molecules like water. And this is what the bilayer looks like. So you can see the extracellular fluid would be up here interacting with the phospholipid heads. Intracellular fluid down here would interact with the phospholipid heads, and the tails are sandwiched in between. Within that plasma membrane, we also have another lipid molecule called cholesterol. Cholesterol is an important molecule in strengthening and stabilizing the plasma membrane, specifically against changes in temperature. So that membrane is a very fluid membrane, but cholesterol helps to maintain its integrity. We have integral proteins and peripheral proteins. Integral proteins are found spanning all the way through the plasma membrane, and they can act as carrier or channel proteins or protein pumps, allowing materials to move through them. Peripheral proteins are found on one side or the other side of a plasma membrane. They're easily removed compared to the integral proteins, and they often act as enzymatic proteins. They might act as a protein that contains a carbohydrate group so that you can have receptors associated with them. It just got dark in here. It's like all of a sudden got creepy. My lights just turn on and off on their own. It's weird. Woo! There's the lights. Sorry. Had to turn them back on. Okay. So let's look at what these structures are. Here we have a glycoprotein. Here we have a channel protein. I told you about glycoproteins, have carbohydrates attached. Glycoproteins can be peripheral proteins or they can be integral proteins. Here you have a peripheral protein. There's another peripheral protein. Here's an integral protein. Here's an integral protein. Integral proteins are the ones that can allow molecules to move across them. Peripheral proteins are not going to be able to do that. And here we have that cholesterol that I was talking about. So what is the major function of the plasma membrane? It regulates materials moving in and out of the cell. It's kind of like a bouncer. So if you've ever been to a hop-in club and you have those people standing at the door and they either don't let certain individuals in or they kick people out because they're getting too rowdy, those are the bouncers. Plasma membrane is like the bouncer of the cell. It only allows certain molecules through the membrane to go into the cell or out, and anything else is going to have to enter through a different means, not through that plasma membrane. So there are different processes to allow materials to enter. Two major ways materials move is through passive or active processes. The passive process is going to not require energy of any sort, and materials will move down their concentration gradient from higher to lower concentration until there's an equilibrium that has been met. And there are two major types, diffusion, two types of diffusion, simple diffusion and facilitative diffusion. And then we have osmosis. And then with active transport, we use energy. So in general, the energy is going to come from the ATP, but there's a secondary active transport where we're using energy of ion gradients, which we'll talk about later on, okay? So this is simple diffusion. Simple diffusion is the movement of molecules from a higher to a lower concentration down a concentration gradient. So here we have some small nonpolar molecule that can go through the plasma membrane. So the plasma membrane is very selective. It allows small and nonpolar molecules to move through them and does not allow polar or larger molecules through. So these small nonpolar molecules can move from higher to lower concentration through that plasma membrane until we have an equilibrium that is met. That's simple diffusion. And I did link this to a video. This is a video. Okay, so I did link it to a video, but the video is not playing right now, so I apologize. If you are struggling with this process, just ask me in class, and I can go through it again. Another thing you can do is just look up, put in your Google bar, in that little Google spot, just put in diffusion animation, and there's a lot of different animations online about diffusion. Facilitative diffusion, then, is the movement of molecules from higher to lower concentration down a concentration gradient, just like passive diffusion, except for with facilitative diffusion, we are moving through a protein carrier or a protein channel. So here's a protein channel. Channel proteins allow molecules to move directly through them. Carrier proteins will grab specific molecules and carry them across. That's how glucose enters or exits the cell, is through carrier-mediated facilitative diffusion. Most of our ions, like sodium and potassium, move across the membrane through channel proteins. Let's see if this one works. This one worked. So this is facilitative diffusion, and actually once you're on this website here that I'm going to be bringing up, you can look at osmosis, you can look at diffusion, you can look at all these different, how diffusion works, so you can look at any of them. So this is a free website. You can just open it up and you can go and look at the different things.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Okay. The third type of passive transport is osmosis. And osmosis is a form of diffusion, but now we're moving water through a selectively permeable membrane. Water can move two different ways through the plasma membrane. So we're going to move water from higher to lower concentration down its concentration gradient, either directly through the plasma membrane, and water is a polar molecule, but it's one of those special molecules that's able to do things that most polar molecules can't. Or what mainly happens is it will go through protein channels called aquaporins. So these aquaporins, the more we have, the more water will move into or out of the cell, depending on which way we're moving, okay? When water moves, it's going to move, oh, and I just said this. When water moves, it's going to move from higher to lower concentrations. It's a diffusion process, but it's going to move from an area where we have less solute to an area that we have more solute. And so here's a figure from your textbook that shows what diffusion or what osmosis looks like. Over here you have you have 7 different molecules, so solutes, and over here you have a lot more than 7. I'm not going to count them, but there's a lot. There's like 20 or so over here, 30 maybe. So water moves from higher to lower concentration. There's a higher concentration of water to solute on this side than this side. So it's going to move until we have an equal concentration of water to solute ratio on each side. And here's your osmosis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay. So let's look at these different terms. We're always going to be gaining or losing water through osmosis, and depending on the tonicity of the solution that our cells are in, we're either going to gain or lose water. So a hypertonic solution is one that's going to cause your cells to lose water. This is a mistake. Hold on a second. All fixed. In a hypotonic solution, you have more water outside of the cell than inside of the cell, and so the cell gains water. Isotonic solutions are solutions where you have equal amounts of water to solute ratios in the cell and in the solution surrounding the cell. And so here's what these solutions do to cells. If your blood is in a plasma that is hypertonic, then water is going to leave the blood and move into the blood plasma. And that's going to cause our blood cells to start to shrivel up. If your blood is in a solution in water or in blood plasma that is hypotonic, then water is going to move into the blood cells, and that's going to cause the blood cells to gain water and become very, very round. That's hypotonic. So I always think of children. Little children are very small, and typically they're very, very active. I have a 2-year-old grandson that's living in my house right now, and he is uber active. And so that's also known as hyper, right? And so hypertonic makes your cells get smaller, whereas hypotonic makes your cells get larger, and hippos are very large. So hippos and hypotonic sound the same. You can think of hypertonic solution when you are dehydrated. When you are exercising, your cells are going to end up losing water because there's going to be more water in the cells than in the plasma. You're losing that water from the blood plasma, and so water leaves the cells, enters the blood plasma. When you drink a lot of water, so if you have a lot of water in your system, very hydrated or overhydrated, that is when your cells are going to take in more water than they normally would. And so they'll have more water than, or they will eventually have more water than they're supposed to potentially. And that can cause the cells to actually burst, and that's termed hemolysis. What we always want is equal amounts of movement of water into and out of the cell, and that is called an isotonic solution. And that's what our blood plasma is typically to our cells. I'm going to stop here. This is a 23-minute video, so that's a pretty long video already. And we'll start in on active transport in our next video.